Okay, so this is the first of my set of lectures on thinking about the mathematics of infectious diseases. That's lecture number 1. This picture will be an accompaniment to all the lectures I give. It will be my first slide. So pay attention to it. There's a sort of story behind it, why we are interested in a picture of that sort. So here are some references. This is a particularly nice book, Keeling and Rohani, and some of the figures that I will take will be from here. This is also a book that has a bunch of programs at the end of it, which are available for free online. So some of the assignments that I would like you to do that are numerical assignments can be simply done by just downloading these. They're Python programs, MATLAB programs, C programs. So something among this should work for one of you at least. And then you can have a look at that, and we can try running it somewhere towards when we have our tutorial session. This is a more recent, more mathematical book, the Diekman Heasterbeek and Britton book. This is a classic. This is one of these big, it's about this thick and has pretty much everything that you wanted to know about infectious diseases of humans. It's a bit dated by now. There's a nice web page, I don't know if it's still up, by Chris Myers at Cornell, which is devoted to teaching some of the things that I will teach you about. If you want a little more mathematical treatment, the SIAM review of Heathcote from 2000 is a very standard reference that does things at a somewhat more, slightly more mathematical level than I will deal with here. This is a book that really deals with vaccines and sort of how to determine the efficiency of vaccines. This is a nice review article and reports on progress of physics. This is a fairly recent one that again covers much of the ground that I will cover. So what I'd like to do is basically make it possible for you to go back to the literature and understand what they're talking about much faster. For example, at the end of these lectures, hopefully you'll be able to pick up this particular review and understand pretty much what it's doing, why, how these calculations are done, what the terminology is, and so on. So why should we think about infectious diseases? Here's a picture that is one example of why infectious diseases are important, and why thinking about infectious diseases is important not just for mathematicians. This is a course on mathematics of infectious diseases, but really for anyone who is interested in health of populations and what determines this. This is an Ebola patient. It's from about 3 years ago. You can look at the extreme care that's being taken to transport the patient. Look, these people are covered top to toe, including having these little booties on top of their shoes. And all of these people here are standing at appropriate respectful distance. And the reason they do this is because Ebola is a disease that's conveyed through body fluids, pretty much any type, tears, blood, etc., feces, and so on. So even very small quantities of that, if it's ingested by these people, they will fall sick and die with very large probability, which is why these extreme precautions have to be taken. Here's a little more current information. So this is as of 4 days ago, so that's 27th November 2018. Those of you who've been following the news will remember that there is now an outbreak in the Democratic Republic of Congo, and these circles roughly refer to the size of the outbreak. Some of these are confirmed cases, some of these are probable cases. This is a report that's taken from the World Health Organization. So in general, if you want to know about diseases, get data about reporting for smallpox and for measles, Ebola, etc., it's a WHO document that you would normally go to. They give you a nice overview. They will tell you where the cases are being reported, how many cases there are, what is suspected about it, what is the nature of the strain, and so on. And over here, between 30th April of this year and 26th November, when the document was prepared, you can see how the number of cases have gone from pretty much just maybe one probable case out here to what you call an epidemic, where the number of probable cases that are reported every day begin to increase as you move. And now the question is it going to increase like this, or is it going to turn around at some point? And ideally, we would not like it to increase like this, because it would mean that these circles would get larger and larger till they cover both the Congo as well as the regions that surround it. So you want to ask, what is it that I can do, and what is it that a mathematical treatment can help me do to understand whether this curve is going up? If it goes up, how far does it go up? If it goes up and then comes down, how fast will it come down? And can I sort of induce it to sort of come down right away from this point? So understanding this problem from a mathematical point of view is important to understanding what you should do to be able to tell you the difference between 10 different ways of approaching this problem. Do you quarantine patients like these? Is there a vaccine for Ebola that you can use? Do you prevent people from going to hospital and say, look, lock yourself in your room, stay there, we will push in medicines through the window, just eat those medicines and you'll be okay. And that's, in this case for Ebola, there is no known medicine that you can actually use. So what is the nature of treatment of a disease like this is another question that we would like to ask. So here's my plan for the lecture. Between the first and part of the second lecture, I want to tell you about the background to disease models. That's basic biology, epidemiology, and a bit of the history of people who've been thinking about models for infectious disease. So this talk will be unusual because there will be no mathematics in this talk. It will only be about terminology. Because later, if I want to talk about reproductive ratio or cohorts or cross-sectional analysis, etc., I want you to be able to understand what is the language that I'm using. And we should not forget, we can be doing mathematics, but we should not forget the underlying problems, which are problems of public health and disease and biology. So whatever the basic terms that we need to come to understand that we will use here in our mathematical modeling are terms that we must really understand at a fundamental and basic level, and also so that you don't look foolish when you go and talk to a clinician or a public health specialist. You must understand the sort of terms that they use and the sort of language that they use and are familiar with. Okay, so that will be my main aim in much of this lecture. Then we will go on to the classic model of infectious diseases called the SIR model. How many of you have heard of this model at least? Okay, that's pretty good. So that's roughly about a third of the people present here. We will talk about things like reproductive ratios, vaccinations, herd immunity, oscillations, and give you some idea of how to solve the model for interesting properties that you might want to look at. This is a simple model. Its importance is precisely in its simplicity, because you can take a simple model and then make it more complicated by putting in all the things that you think you left out the first time. So you would want to look at making it stochastic, you want to look at making spatial dependence on it, and then asking what happens for more complicated diseases. For example, diseases that are vector-borne, mosquito-borne, for example. That's what we'll cover a little further up. And then finally, as Shitabra mentioned, I want to talk a little bit about network and agent-based models for infectious diseases. So this is in some sequence between simple models, more complicated models, and really sort of state-of-the-art ways of thinking about this problem nowadays. You with me so far? Have we been clear? Okay. So what we want to think about is a field called mathematical epidemiology. And the word epidemiology, epi means on, demos means people. These are old Latin roots. This is the study of what is upon people, which is concerned with the dynamics of health and disease in populations. So here's an old plaque commemorating the plague, when the plague actually swept through the UK in 1348. And this killed 30 to 50% of the country's total population at that time. And I'll come to some examples of how devastating plagues like this can be, devastating infectious diseases that have a high mortality can be. Because 30 to 50% is basically half the people in this room. You won't see it over the next 3-day period if you were all exposed to plague. What we're interested in is, since we're talking about populations, if you imagine this to be a pie chart of the population, a good fraction of the population enjoys what we might call good health, and there's some fraction that enjoys what we might call ill health, they're unwell. Even this population is a function of time. Their health can fluctuate. For example, people can become better or worse or better or worse. So it's some complicated curve that describes what fraction of the population at any given time is ill. Okay? That can change with time. So what we want to do in epidemiology is to identify the distribution, where the disease is, incidence, the number of cases of the disease, and etiology of human diseases to improve understanding of the causes of disease and to prevent their spread. And this word etiology is a little more of a medical term. It's a study of causation. What causes a disease? So before we get to diseases, it's important to try and understand even very basic terms that surround them and how they have evolved over the years. So I just want you to think about some of these terms because these are issues that are larger than the issues that I will cover in these lectures, but they're important to think about. So if you look at the old WHO classification of what is health going back to 46 and 48, It says it's a state of complete physical, mental, and social well-being, not merely the fact that you're not sick. There's a little more than that, and it also has a component of mental well-being. It's not just physical well-being, but also mental well-being. Some of you may know that depression, I think this year is a sort of year of mental health declared by the WHO, and depression and associated psychological problems are very important in this country. This is an extremely underserved area of illness, and the WHO definition recognizes that. What is disease? And the reason you must be clear about this question of what is disease is because if you have some limited, none of us, no government, no community has an unlimited amount of money. So at some point, governments must make choices, what to treat, what not to treat, how much money to put into a certain disease and how much not. So you must first know what is a disease, does it qualify as a disease, and which diseases are most worth the investment of time and money. Are there diseases that are either untreatable or treatable at such large cost that all your budget will go in treating 2 or 3 people? Or should you concentrate on simpler things, for example, like improving health and sanitation, by which the quality of life and general state of health of a much larger population would improve? And these are decisions that governments and civil society and people who think about policy must make. Now, if you ask somebody, are you ill or are you well? Their own definitions will change. It depends upon their class, gender, ethnic group, and factors as proximity to family support. In general, women, for example, might be a little less likely to say that they're ill compared to men because they have to shoulder a large fraction of home responsibilities. Also, what counts as a disease has also changed with time. What we thought was a disease 20 years ago may not be a disease now. But usually it goes the other way. So here's one example. So osteoporosis, which is a condition which as you grow older, the architecture of your bone changes and you go from sort of being straight up and rigid like this to a situation which you see many old people. They're bent over. It's a consequence of osteoporosis. This was recognized as a disease by the WHO in 1994. But before that, we understood it as a fairly common cause, or not cause, or fairly common consequence of normal aging. So you've converted something that should happen under normal circumstances if you live long enough to something that you now consider as a disease and worthy of being treated. You can ask the following deeper question. Suppose someone has a genetic predisposition to a disease. For example, suppose you carry a mutated version of a gene that makes it more likely that you will have breast cancer in later years. Is someone with such a predisposition already ill? Do you count them as being already ill? Or do you not do that? So what you choose to be a disease model as a diagnostic criteria have very fundamental questions or debates about ethics, human rights, and to what extent you're willing to tolerate differences, for example, between this person and this person. Is this person really ill? Or should they say that, look, all of us when we grow old, we will have problems that are associated with old age. These are not diseases, but a natural progression of the human condition. So let's get to what is disease. A disease, at least we can say, that is some abnormal pathological condition that affects all or part of an organism. And sometimes people use the word disorder, and disorder is just some abnormality in a function or some disturbance in regular function. Often we use the slightly more colloquial words illness or sickness as synonyms for disease. And sometimes people say that, look, I feel ill. It's more relates to a patient's personal experience of his or her disease. So it's clouded by what the patient's own threshold for pain and discomfort is. Someone may be very sensitive to small amounts of pain and they say, I feel terrible, I don't want to go anywhere. And someone may have a much higher threshold level. So that's really the personal experience of the illness and not the sort of static fact of the disease itself. Now that we've talked in general about thinking about health, thinking about disease in a much broader context, let's now zoom in on the question of what are infectious diseases. Okay? So infectious diseases are mostly, very broadly, and I'll tell you some specific counterexamples, are caused by microorganisms. And the microorganisms that are involved are such as bacteria, viruses, parasites, or fungi. If a bacterium, for example, or any agent causes a disease in man, it is said to be pathogenic. So that's one important term that we will use. The other useful word is a reservoir. A reservoir is some natural environment in which a pathogen typically lives. For example, it may naturally live in a human being, it may live in an animal, or it may live in some environmental component such as water. Okay? So that's pathogenic, that's reservoir. Infectious diseases are infectious because you can transfer them from one person to another. That's what the word infectious means. You can either transfer it directly, where the disease-causing organisms such as virus or bacteria pass directly from person to person. I may have a cold, I may cough here, and 2 days later you may fall ill. So that's a sort of direct transfer between me and you. But it's also possible to transfer it through an intermediary, which is a vector such as an insect. For example, dengue is transmitted through the bite of a mosquito called Aedes mosquito. So if I suffer from dengue, I can be bitten by the mosquito. There will be a life cycle of the parasite within the mosquito, and then the mosquito will bite someone who's free of dengue and give them dengue. So that needs some intermediate quantity to transfer the disease from person to person. So here are some examples. Colds, influenza, and chickenpox are all examples of infectious diseases. So that's what they look like. These are what bacteria look like. Bacteria typically tend to be small. They are about, roughly speaking, let's say 1 micron. 1 micron being 10 to the power minus 6 meters, which is about 6 orders of magnitude smaller than a meter, which is roughly this size. So it's really small. Viruses tend to be smaller, although they come in a bunch of different sizes. So maybe this is about a tenth the size of this, roughly speaking. These form multicellular aggregates, so they're fairly large. Protozoa can be anywhere between about 1 to about 20 or 50 microns in size. So cholera is a bacterial disease. H1N1 is viral, dengue is viral, malaria is parasitic, HIV/AIDS is virus, chickenpox is a virus, flu is a virus, tuberculosis is a bacterium, MERS is a virus. This is Middle Eastern Respiratory Syndrome. Ringworm is a worm. What did I call it? Okay. A fungus. Candidiasis is a fungus. Malaria is a parasite. Amoebiasis is also a parasite. Okay, so you can see that there are many different types of pathogenic organisms, but at least at this level I can calibrate them or divide them into bacteria, virus, fungi, and protozoa. So there's another word that is used a little bit, and if you look at this Keeling and Rouhani book, they will use the terminology microparasite and macroparasite. The microparasite, they refer to things that are sufficiently small, such as bacteria and viruses, and these multiply fairly fast inside a human. So you can ignore the dynamics within the host and concentrate on the nature of the host itself, rather than the bacterium or the viruses. These are more complicated. They have a long and complicated lifestyle. You can have a ringworm that enters you and then stays with you for a long period before it actually becomes you are actually conscious of its presence. And vectors are a different sort of sequence of transmission from either the microparasite or a macroparasite. So here are different types of vectors. As I said, mosquitoes are involved in dengue fever, yellow fever, chikungunya, malaria, all of these really fairly miserable things. Ticks are responsible for Lyme disease, spotted fever, sandflies for an important disease in India called leishmaniasis. Tsetse flies more, and this causes sleeping sickness in Africa. And fleas, of course, are responsible for the Black Death, transmitted by fleas which live on rats to humans. Snails called schistosomiasis, black flies called river blindness. All of these are vectors that act as intermediates before a person can be infected with a pathogenic organism. Having told you about infectious diseases, let's not forget non-infectious diseases. And these are diseases that are not due to disease-causing organisms. So one example would be genetic diseases such as Down syndrome, hemophilia. And there are other examples, for example, that come from lifestyle or environment, such as cardiovascular disease and skin cancer, as well as diseases involved in disruptions of the body's own metabolism, which are diabetes. So just remember this, infectious or communicable is caused by bacterium, virus, parasite. We didn't discuss misfolded proteins, but that's another source of disease here. And really the bottom line is other people can get them from you. Over here we have non-infectious, non-communicable diseases that come from these causes here, and other people cannot get them from you. And we will be worried about this column here on the left as we go on. So here is a bunch of the non-infectious diseases to add to the infectious one: hypertension, cancer, hemophilia, cardiovascular disease, obesity, etc., on that side. Having said all of this, Now that we know that microorganisms, bacteria, viruses, protozoa, etc., are responsible for making you sick with an infectious disease, he has introduced another term which is called the chain of infection. And the chain of infection refers to the whole process by which an infectious organism living normally in its reservoir enters through some mode of transition into the human being, and then enters at this point, enters what is called a susceptible host. The word susceptible will come up many times. Susceptible is one who is about to fall sick or who could potentially fall ill. And so you can see that it involves both the organism as well as the host. So the interaction of the chain of infection itself involves some interaction between both host and the pathogenic organism. And the whole issue of the interaction between the host and pathogen has a whole bunch of different complexities. Is the host successful at all in making you sick? That depends upon the susceptibility of the host. How efficient is the pathogen at transferring from person to person? That depends upon modes of pathogen shedding. Where is the pathogen found? Is it found in water? Is it found in air? Does it have some special, is it sort of identified with bats who live in dark, damp caves, etc.? That's the nature of the reservoir host over here. How does it enter the body? If I wear a mask over my face and only boil my water, am I fairly confident that I have at least circumvented some routes of entry and changed the nature of modes of transmission of that particular? Okay. Having told you all these terrible things about how, dengue, chikungunya, cold, this, that, etc., it's reasonable to ask the question, why aren't we all falling sick all the time? It's not, I'm sure all of you know someone who's had dengue or malaria within the last 3 months, or a terrible cold, or H1N1 or something like that. It's pretty much unavoidable. In fact, we've had 3 people who were supposed to come, somebody fell ill with the typhoid and couldn't come, and someone else and couldn't come, etc. So that raises the question of what keeps us okay. And the answer is that it's our immune system that protects us, and it protects us in a bunch of different ways. So all of these flu, cold, disease, etc., is trying to attack this, but the immune system defends the body. And here it's where any prior contact with the virus or bacterium that helps the system recognize the invader. And that's why vaccinations are important, because vaccinations provide a little key to recognizing anything new that comes along. So that you can say, even though I have not been infected before by the bacteria or virus, I'm able to recognize the foreign element that is presented by the bacterium or the virus, and then my immune system can be ready to deal with it when it actually comes. So a little bit about the immune system. So if you'll realize that I'm just doing sort of stream of consciousness here. I'm telling you about a bunch of things, not in great detail, but things that you need to remember about the complexities of thinking about disease and how disease is transferred from person to person. So the first line, you can think of these as different lines of defense. The first line of defense against a pathogen is what is called non-specific physical and chemical defenses. Your skin is a good barrier because the skin is covered with a layer of dead cells, and the cells prevent entry. So only, you can get tetanus if you have something that penetrates through and hits your blood at some point. Your saliva contains various enzymes that help to break down bacteria. So it's actually not a bad idea to wet your finger and rub it on something if you've got cut. It's a very mild antiseptic property that it has. So does your mucous membrane. Your stomach acid, for example, is strong enough to be able to degrade some number of microorganisms that can't stand a strongly acidic environment. And of course, coughing, sneezing, vomiting, diarrhea, etc. are all ways of getting rid of various pathogens that may have entered you. There's one part of immunity that is called natural or innate immunity. That is, you don't need any prior exposure to something foreign in order for those defenses to be activated. And these are associated with cells called macrophages, neutrophils, eosinophils, basophils, etc., and something called the complement system in immunology, which we won't talk about. And this last is in a very high level and very important and interesting part, which is acquired immunity. And that refers to the fact that once your body has been attacked by a pathogen, the next time around, either the same pathogen or a new pathogen, it's able to recognize and mount a defense that is very effective and fast to basically clear that pathogen out completely. So which is why you're immunized in early life, and also which is why you catch measles as a child, you typically won't sort of worry about catching it again at later point. Because your body now recognizes the fact, it recognizes the organism that gives rise to measles, and is able to deal with it if ever. So you can be happily in a room with 20 measles patients, but once you've already got it in the past, you're protected against it. Okay, and that's the job of the adaptive immune system, which is acquired immunity. So as I said, we're going to look here between innate and adaptive. Innate defenses are surface barriers, internal defenses, but the adaptive defenses come both with B cells and with something called T cells. Okay, so the sort of reason I'm telling you all of this is that the immune system is an extremely interesting object on its own. So there are two words that you notice here, humoral immunity and cellular immunity. The humoral immunity is mediated by molecules that you find in extracellular fluids such as blood. They're antibodies, complement proteins, antimicrobial peptides, etc. But the cellular immunity comes from cells themselves going and grabbing on to pieces of invading the other types of cells that form and that are invading it, little pieces of protein that it recognizes, and that comes from cellular immunity. So here's a quick cartoon of an infected cell infected with various pathogens, and once these pathogens attack the cell, or even if they're present in the bloodstream, that triggers an innate immune response. So this is an immediate response. It turns on just like this. But the learned response is delayed. This is the acquired response here, and that involves the T cells and the B cells that I told you about. The B cells make antibodies that target very specific pathogens, that is, this little stick-like object here can recognize the shape of these pathogenic molecules that are coming in. These T cells kill the pathogens and attack, so they engulf these infected cells and kill them, and some of them become memory cells that retain the memory of what happened in an earlier infection for a later point. This is the only slightly technical biology slide, and this refers to the fact that all of these different types of cells, the words that I mentioned to you, the erythrocyte, the mast cell, the neutrophil, the eosinophil, the plasma cell, the macrophage, the T cells, etc., all of these derive from what are called stem cells. Okay, these are called hematopoietic stem cells that give rise to all of these different types of cells through a process of differentiation. So the word hematopoiesis is the process by which all mature blood cells are produced. So all of these are different types of blood cells. And these stem cells can give rise to all of these different types. The reason I mention this is also because this is an extremely complicated picture. How is it that one single cell here knows that it can give this, all of which are biologically different? The sort of nature of the markers that these cells carry are different. The functions are different. How does this single type of cell here, the hematopoietic stem cell, know how to make all of these different types of cells? And how do these cells know what their functions are supposed to be? How is it the T lymphocyte knows that its function is to attack infected cells? How does the B lymphocyte know that its function is to present antigens on its surface so that it can attack, look for certain protein molecules that are presented by invading cells? So just to make the point that this whole field of immunology is a very complex and beautiful field. And you really need more mathematical studies in this field. This is not what we'll be talking about, but if any of you are interested in looking at problems in the future, in the field of mathematical biology, here is certainly an area that I would suggest that you thinking about mathematical immunology is really going to be a very important field in the next 20 or 30 years. Okay, so right now we know what is disease, what is health, where do we get diseases from, bacteria, viruses, etc. So we have some of that history in why do we not fall ill? Because our immune system helps us. What are the parts of the immune system where we have sort of external physical and chemical barriers, we have innate defenses, we have acquired defenses. We know a little bit about it. At least we know enough to go and read a particular book that deals in detail with some of these things. So let's go on to a little bit more about other parts of infectious diseases. And that's a historical perspective on where these diseases came from and where did they start. So pretty much as long as there have been humans, there have been diseases. And so the records go way back. Even in ancient Egyptian Chinese writings, they describe smallpox. And although we don't think about smallpox now, it's effectively been eradicated. It's just in one lab, maybe one lab in the US and one lab in USSR under extreme safety conditions. It may have been responsible over the years for more deaths than all of the other infectious diseases combined. There's some evidence that malaria and polio have existed since ancient times, and leprosy may also be a very, very old disease. Ancient Greece and Egypt reports of epidemics of smallpox, leprosy, and tuberculosis. There was an epidemic in 430 BC in Athens, which killed about half the population. Interestingly, it's not known what was the causative agent here. Was it influenza? Was it a type of Rift fever? But its likelihood is that that particular agent doesn't exist now. It's sort of gone extinct, which is also an interesting thing about the microorganisms that they change with time. The collapse, epidemics of disease can be enough to collapse whole empires. So the Han Empire in China in 3 AD may have collapsed due to plague and may have even led to the end of the Roman Empire. And remember the first sort of picture that I showed you of the plague, which killed roughly 3 Europeans out of 10 and roughly 50% of people in Britain. So people have also weaponized diseases. For example, the Aztecs were a community in this part of South America between the US and mainland South America, was really made easy by the fact that the invaders who came, the Spanish invaders, brought smallpox with them. And the Aztecs had never seen smallpox before, so they had absolutely no immunity, which is why immunity to a disease is very important. And so this is something, there's another piece of history that is also repeated when the Americans when the first bunch of people, I don't know if you call them invaders, from Europe went into America and decimated the native tribes. One way they did that was to take blankets which had been used on smallpox patients and give it to the tribes. And again, those tribes had no existing immunity at all to the disease, and large numbers of people died. So you can also use diseases as weapons, not just as an example of bioterror. This syphilis also goes very way back. It's a highly contagious venereal disease that very likely most European kings suffered from at some point or the other. This is a history that you may not know of, which is the 1918, 1919. This is still within your grandparents' time. And this was at the end of World War I. And this is probably some type of H1N1 flu. In India, the estimates are there were 15 to 20 million dead at that point. And of course, more recently, you know of SARS, bird flu, Ebola, chikungunya, dengue, and Nipah. All of these have a significance that relates to the first picture that I showed you on my cover slide, which had a bat and a bird and something in a hand. All of these are what are called zoonotic diseases or zoonoses. They're caused by infections that are shared between animals and people. Okay, so the bats were certainly an important group. The bats, the birds, etc., etc. These are all viruses that normally circulate in those animals, but somehow make the transition to humans. They acquire some mutation that makes them deadly to humans as well, and then they cross over. So, given the fact that pretty much most of what you will read in the papers now has to do with one of this, either you read about dengue or SARS or Ebola or Nipah virus more, it's important to understand this new source of zoonotic diseases and to understand where new viruses or emerging infectious diseases can come from. Okay, so that was a very quick survey of the history of infectious diseases, how far they go back, and why it's important to know that there is a long history to these organisms that have been affecting people. And much of our progress really in understanding how to deal with them has been in the last 200 to 300 years, but they have some precedent in what we knew about them even as much as 1,000, 2,000 years ago. So let's talk a little bit about where medicine came from. Of course, people have used medicinal plants since about 2,000 years ago, and Indians were very important in that. So Sushrutas did the first surgeries, learned how to pull teeth, set broken bones, identified more than 1,000 diseases, as well as a large number of medical drugs. So in India, surgical instruments, eye surgery, rhinoplasty, which is sort of changing nose surgeries essentially, go back way beyond. And these people also wrote about what they did. So the Sushruta Samhita and Charaka Samhita are classic old works that describe what Indians 1,500 years ago, 2,000 years ago were actually doing in terms of medicine. What they did then still has echoes now. And it's interesting to ask that if you go back to natural products and traditional medicines, can you use them as sources of new antibiotics? Are there curative elements inside that were known, and even though the curative agent was not understood at that point, is it possible to go back to them to derive new types of medicines that may be useful in ways that we had not anticipated? And one example, of course, is the drug called Taxol, which is an anti-cancer drug, which is derived from a natural product, which is the Himalayan yew. It's a type of tree from the bark of the Himalayan yew tree, etc. So there are meetings now that actually look at this interface between traditional medicines and modern medicines. Of course, as the Egyptians faded, the Greeks arose. And much of what we think about as modern scientific medicine, really, there's a direct line from the Greeks to us. And Hippocrates is very important in the history of medicine. He founded a school of medicine, the Hippocratic School of Medicine. And he really did a very systematic study of clinical medicine, actually introduced the direct examination of a living patient as a way to understand disease. And of course, you've heard of the Hippocratic Oath that every medical professional must take, that sort of sets the bounds on the way they should interact with their patients. There are other schools of medicine. For example, traditional Chinese medicine is about, again, 2,000 to 3,000 years old. It's contemporaneous with Indian schools of medicine. Long ago, they knew how to take a pulse. They had medical schools right at that time, and going way back, I'm sure it was as hard to get into them then as it is now. They're very old records of smallpox, use of vaccinations, etc. There's some complicated sort of spiritual stuff attached to it, yin yang, 5-element theories, etc., etc. And typically, if you go to a traditional Chinese medicine person, you would see acupuncture, herbal medicine, various types of physical exercises. And in a sense, this is probably not too different from the way we in Ayurveda approach the same sort of thing. There are various complicated explanations that we have for where Vada, Pitta, Kava, etc., etc., etc. These are all sort of traditional medicinal approaches that may not be grounded so much in what we consider now as modern medicine, but there are parts to it that might be interesting and useful, and you shouldn't throw it out altogether when you think this is old stuff, we don't know what it is, we know exactly what we're doing now. Let's come to more modern ages. So it's Vesalius who dissected human beings and published pictures that actually looked like this, that identified muscles, bones, etc. Harvey discussed blood circulation and understood that. Montagu was an aristocrat who did a huge amount to actually encourage public understanding and acceptance of inoculation against smallpox. And Jenner, of course, introduced the smallpox vaccination at that point. So now let's come, so that's again a quick survey of the history of medicine. Let's now come to a little more modern understanding of bacteriology. And that's when you cannot avoid these two people, Louis Pasteur and Robert Koch. Pasteur is French, Robert Koch was German. It was Pasteur who founded the germ theory of disease and said that, look, ultimately there must be some small microorganism that is present, that conveys the disease between human beings and to human beings. He developed vaccinations for anthrax and rabies, and Koch was a physician about 20 years younger than Pasteur. He won a Nobel Prize in 1905, that's what he looked like. They didn't like each other at all. And there was, part of it was just pure scientific competition between them. They didn't want the other person to do well at all. So there was a lot of, horrible intrigue that went on at that point. Koch showed that you could transmit anthrax from one mouse to the other. He found bacteria responsible for various diseases over here. These two are known as the founders of modern bacteriology. But these are important because these are what are normally used when you think about identifying the cause of a disease. So Koch introduced what are called 4 postulates that determine whether a specific disease is caused by an agent, a microorganism. So what's in here are various sort of caveats to what Koch originally wrote, and these are not as important now as they were 50 to 100 years ago. But the first point is that there must be a specific agent that is associated with every case of the disease, but it should be absent healthy organisms. Okay? That sounds reasonable. There should be some microorganism that's found if you're ill, if it's associated with that illness. But if you're not ill, then there shouldn't be any trace of that in your blood, for example. Of course, that ignores the fact that you can have something called an asymptomatic carrier, which I'll come to later, in which the person bears the microorganisms within themselves, but they don't show any outward signs of the disease. So they're carriers. They can transmit the disease, but they don't fall ill themselves. The second postulate is that it must be possible to isolate the agent from a disease host and grow it in culture. Okay? And the counterexample to that is what I said is misfolded proteins or prions. And there are a certain set of diseases called prion diseases that come from this. And those certainly, misfolded proteins you cannot grow in culture. It doesn't make sense to do that. It's some specific combination of cellular events that lead to that misfolding. And once they are misfolded, presumably they set a template for other proteins to misfold. And diseases, for example, BSC, the bovine spongiform encephalopathy, is an example of a prion disease. The third point that he made is when you take culture-grown agent into a healthy host, the agent must cause the same disease. Okay, so that's sort of saying that it's a microorganism that is actually responsible. However, we know that not all exposure leads to infection, so there's a little caveat there. You can be infected, but things going on in your own physiology may ensure that you are not affected. You're able to shrug that infection off. And finally, you should be able to take the same agent from this infected host and do the same things that you did out here. So there are caveats to this, but nevertheless, it's important to recognize that these are very fundamental to the way even now we think about disease. There is an agent, it's an external agent, it's present in the person. You should be able to isolate it, you should be able to grow it in culture, you should be able to take those cells, put them in somebody who's healthy, and that person will then fall sick. So this informs the way we think, even though we may not believe the details of this so much more nowadays. And we're now aware of many more counterexamples to these ideas. Here are vaccinations and the whole sequence of vaccinations. So certainly the BCG, polio vaccine, these are things, the MMR vaccine here. These are all examples of vaccinations that are very important to our own health. Here's an example of, this is the number of deaths per year by measles, whooping cough, diphtheria and scarlet fever. And the introduction of antibiotics that came in here sort of sets this decline. And you can see how the introduction of whooping cough vaccination, introduction of diphtheria vaccination and measles vaccination has led to essentially no deaths per year by the time you get to beyond about 1960. And you can also look at Australian data when you introduce vaccination at this point here. So over here it sort of peaks and it sort of looks as though it's sort of stabilizing at this point. But now it begins to come down, and by the 1960s here, there are no deaths anymore. And for polio, this is the introduction of the Salk vaccine, the introduction of the Sabin vaccine here. And again, that number is pretty much zero. And now, apart from a few cases occasionally reported in the Northwest Frontier province or in parts of Africa, there are no more cases of polio. And hopefully, in the next 10 or 20 years, we'll get rid of it completely. Okay. So that's again a quick view of something else. It's a quick view of vaccinations. So let's talk a little bit more, let's focus a little bit more on things that have more to do with mathematical treatments of disease. And that's about epidemiology. So the first people who looked at causes of mortality, why are people dying, where are they dying, etc., is a person called John Gaunt and William Farr. He was the first person to look at mortality data, patterns of birth, death and disease. Are there differences between male and female? Are there seasonal changes in people falling ill? And Farr is someone who's known as the founder of medical statistics and basically looked at who's dying when and where and why. And he introduced a system of reporting to public health authorities and various basic practices that are good. So these are people who set into place good epidemiological practices 400 years ago. This gentleman here, and there's an interesting story, is John Snow, who's known as the father of field epidemiology, and he used epidemiology to make hypotheses, to test hypotheses, and this was applied to something called the cholera outbreak study that he did. And I'll tell you a little bit about that. He came from fairly poor background. He was the son of a coal yard laborer. He was a bright student and was around during the time of the cholera epidemic of 1848 and 49. And he identified water pumps as sources for cholera. How he did it is actually fairly interesting. So this is a map of a part of London. Each dot here is a reported case of cholera. Okay? So wherever that person was staying was basically put a little dot over there at that point. So if you look at this, you can sort of see something, and it's easier for you to see because you're further off. You can see that there are more dots along here. There's a place with no dots at all, which is here and maybe a little bit here. And the things that are marked here are a pump, a pump, a pump, a pump, and so on. So he looked at this and he said that, look, maybe this is saying the fact that these cases, these dots seem to cluster around this particular pump A on Broad Street suggests that this pump may be the source of cholera. Remember, at that point, they have no idea what caused cholera, how it spread from person to person. So this is purely a descriptive approach to the data that was shown here. So he recommended to the public health authority that they shut down this pump. And they did, and miraculously the cholera epidemic went away. But that raises the interesting question of what happened here and here. Why are there no cases there at all? The reason for that is that the Guinness Brewing Company owned these over here, and they supplied water from a completely different water source. They supplied water and probably also beer and ale, etc., etc., the inhabitants, the people who stayed here. So these people did not interact with this pump at all. So all of these, it's like detective work of going back and putting these dots in, finding out where they came, can we do something, can we shut this pump down? So that's what really is an example of descriptive epidemiology applied to this problem. So he concluded that the Broad Street pump was a source of water. The 2 blocks that were unaffected had their own source of water. He removed the pump and the outbreak ended. And all of this was done without knowing that there was a microorganism involved. Although he finally figured out that water was some vehicle for transformation. And this is one example of how you can take epidemiology and convert it into actual policy. So let's now look at some terms that are useful to us. And the first useful word is something that you will again come across in your reading. There's something called the epidemiological triad. Triad means 3 things taken together. So this is a traditional model of disease causation, and it says that diseases must have 3 components. They must have an external agent, that's your microorganism. They must have a susceptible host, that is some human being who is capable of falling ill and has no prior exposure or immunity to that particular microorganism. And some environment that brings the host and the agent together so that disease occurs. In the Broad Street example, that environment was water. So it brought the agent, the Vibrio cholerae together with the human being who drank the water. So that's the third part, the environmental part. So sometimes this is drawn like this, where you have agent here, host here, environment here. And here are a bunch of factors that influence the response of the host, the response of the environment, etc. For example, age, gender, genetics, behavior, smoking, sex, work, immune suppression, vaccination. All of these are properties of the host. In the environment, social crowding, are you in big crowds where people can cough and then you can sort of fall ill, being exposed to the viruses that they emit? The public health status, are you in an urban or a rural setting? What is the nature of the climate? And finally, with the agent, how efficient is the agent at eliciting an immune response? How pathogenic is it? How infective is it? What is the sort of immune response that it actually generates? Here's a term called the natural history of disease, and that's a course that a disease takes in individual people from its onset, that is the point at which you are infected here, to the point at which you either recover or you die. Okay, so that's a natural history of disease. It's a good term to use. Here's another term that's called a population. Population is some collection of individuals that have some particular characteristic in common. For example, all of you are a population. The common characteristic that you share is that you're all sitting in this room. Okay? The frequency of occurrence of a disease in a population is governed by a large number of different aspects. These are called determinants. And sometimes it's possible to manipulate these determinants and reduce the frequency with the disease in question occurs inside a population. For example, I can say this entire population, I insist that you have only bottled water made by Vesalius or something like that. Therefore, reducing your chances of getting an infection that is transmitted through water by having presumably what is pure water. Depending upon what it is that you're studying, you can either have a very large population or a very small population. For example, you might wish to study a particular disease in a particular cattle population in a particular country. So here you could either consider all cattle in the country, or all dairy cattle in the country, or all dairy cattle of a certain breed in the country. It depends upon what question you want to ask in the context of a specific disease that you want to study. Here's another useful term, and that's called a population at risk. And that is, take the total population and identify out of that only the population that is at risk of a particular disease. For example, if you wanted to study the carcinoma of the cervix, you would remove all men from that total population, and you would look at women who are between the ages of 25 and 69 years, because typically this carcinoma doesn't affect 70 plus years, it doesn't affect 0 to 24 years. Okay, it's this particular subgroup of the total population that is interesting to you from the point of view of this particular disease. Okay, There may be other diseases in which this whole circle is of importance to you. So this defines a population at risk, that fraction of the population that is capable of acquiring the particular disease or disease characteristic that you're studying. So here are the determinants, and usually, even though genetics and environment are important, it's behavior that is the leading determinant of whether an individual falls ill or not. So there are intrinsic determinants that determine physical characteristics of the host or disease agent or the vector, and these are usually determined genetically. Extrinsic determinants are associated with the environment over here. And also you can include interventions made such as drugs, vaccines, quarantines, etc., etc., etc. So now I want to again go a little bit into the types of epidemiological studies. And then at the end I want to talk about why all of these different names and nomenclatures are important and why it's important to know what these terms are. The reason is that if you want to model a disease, you will have to figure out how to get the data that correspond to that particular disease. In what form is that data reported? Who has that data been collected on? Is it the population that you are interested in, or some uninteresting segment of the population that bears no resemblance to the larger question that you want to actually tackle? So let's say a little bit about epidemiology. There are different types of epidemiology. One, in the example of John Snow that I gave you, is descriptive epidemiology. He doesn't draw inferences, he just plots things, looks at trends, and tries to describe what is happening rather than try and find mechanistic reasons or explanations for it. Analytic epidemiology is a little like what we will do in our mathematical treatment a little later on. We want to try and analyze what happens in the population when it falls sick from a somewhat more statistical or analytical point of view. And finally, you have experimental epidemiology, which is an example where you might take a population, and half the population you might want to administer a certain drug against a disease, and half of it you might want to give something that is just a placebo, that is not that, so that you can have a true comparison to understand the effects of that particular drug. So descriptive epidemiology collects data basically that describes the disease under study. It says who is affected, where did the disease happen, over what period did the disease occur. Analytic epidemiology looks at the disease to determine what the probable causes are. And here these studies are usually cross-sectional. All the data is collected at one time. And this provides information about prevalence. I'll define this term, but prevalence means the number of people with that disease at a particular time. All the people. Finally, experimental epidemiology is the design of controlled experiments to test hypotheses. For example, a clinical drug trial is an example of experimental epidemiology. Blind studies, double-blind studies. So blind studies and double-blind studies refer to the fact that even the person administering the drug doesn't know whether that's a working drug or just a sugar pill, just a fake. Because that's really, otherwise the person might be tempted to give the drug to people they know are more seriously ill and to not give it to people who are less seriously ill, thereby biasing the effects of the drug. So when the investigator has some control over the distribution of the determinants to be studied, these are called cohorts. So you select people to be part of a particular study. That selection of people is called a cohort. Cohort comes from the old Roman word, so these are people dressed up in Roman armor and so on. So here's one word that I introduced a little earlier. It's called a prospective study. And a prospective study looks forward in time. It says, all of the people in this room, I'm going to track you as you move forward in time. Look at your body weight, look at your height, look at your other health indicators, etc. Look at your basal metabolic rate, look at your blood sugar and so on. But I'm going to track you as we move forward in time. A retrospective study says that, all right, all of you are here in this room. I want to know what happened to you a year before this, 2 years before this, 5 years before this, 8 years before this. So prospective is forward, retrospective is backward. And if some of you are ill with a particular disease and some of you are well, I want to go back in time to ask what were the circumstances for the ill people that distinguished them from the well people? Is there something that I can find in their previous history that will tell me why you fell ill? So cohort studies are also called prospective studies. As I said, a cohort is just a group of people that are identified. And one way of doing this is to study a particular determinant, divide the cohort into two pieces, and look at the determinant in both of them. So one have a control and then look at a determinant here and then see what happens over a frequency of time. And often there are very large cohorts. For example, in Taiwan, the health screening project has 600,000 participants. Something in the US at the MD Anderson, the Mexican American cohort is about 24,000 participants. So there's 24,000 people who you track on a monthly or a 6-monthly or a yearly basis to find out exactly how their health is changing. So I said, prospective is to look at over time what happens to people here. For example, if you have an obese and lean population, examine them as you move across in time. And the other way, of course, retrospective is to move backward here. Look at desk employees or tire manufacturing workers and ask, if I start with them here, what happened to them? What were the different conditions that they were exposed to 10 years, 5 years from before? Cross-sectional studies are studies that are done at a particular time. And here, for example, a cross-sectional study would be exactly a study that happened either here or here. So prospective here is take a cohort which is a part of a population. Some of them are exposed, some of them are non-exposed, and you track them forward over here to find out what actually happened to them. The retrospective studies are also called case control studies. We won't go into this, but I just wanted to mention the name case control because I won't actually use it over here. But here's a quick diagram that tells you. The cross-sectional study is a study that happens at one point of time here. Prospective studies happen from here and move forward. Retrospective studies happen from here and move backward, identify what happened in the past to lead to a present situation here. These are also called case control studies. And randomized trials are an example of a prospective cohort where you introduce some medication to part of the population, some treatment here, and then you ask what happened to them as you move forward in time. Okay, so now come to the last few pieces of terminology that we'll actually need. The first is an epidemic disease. So we will devote ourselves in these lectures to studying epidemic diseases. And these are diseases that happen in a population excess of its normally expected frequency of occurrence. That's not to say people don't fall ill with something or the other. There's always some background amount of cases of dengue, cases of chikungunya, etc. The concern is when that number increases by 5 times or 10 times, because then you sort of worry that you have a public health emergency on your hands. So it's not this, the baseline need not be 0. The baseline may be some steady, flat level which you're perfectly prepared to treat. But it's large increases above that level that could potentially give rise to an epidemic. Usually in epidemics, you see disease events clustered in time and space. If you go back to the old picture that I showed you of Ebola in the Democratic Republic of Congo, the circles that I drew, it's not the whole state of Congo was not covered in red. There was a circle here and a circle here and a circle here. That suggests that they were clustered in space, but also in time, because you know the number of cases went up and sort of seemed to go up a little bit. We still don't know where that curve is actually going at this point of time. Provided a disease occurs in excess of the expected frequency, you may call it an epidemic, even though actual numbers may be small. If you expect 0 cases, then even 10 cases or 20 cases is a large number. If you expect 500 cases, 510 cases is not such a big increase over that limit. The word pandemic is useful. You would have a word that you will come across. Pandemic is an epidemic that affects several countries. As long as Ebola remains confined to one country, the Democratic Republic of Congo, you won't call it a pandemic yet. But when you have Ebola patients start turning up in the countries that border the Congo or even further up, that's when you will call it a pandemic. A sporadic disease is something that sort of comes and goes, etc. That's not somewhere in between an epidemic and not having a disease at all. It's even when you have a sort of low level of disease and you have a little peak here and then it comes down, a little peak here, it comes down. So that's, again, this is the term that is sometimes used but not sort of very popular in the literature. Okay. Are you with me so far? You're not all asleep? So now let's just get to the beginnings of mathematical models. And as I said, this talk is just going to give you background. We will actually do mathematics later. It is interesting to know what the background is. So the first mathematical models can be traced back to this gentleman called Bernoulli. And Bernoulli was asking a very specific question. He said that, look, suppose people did not die of smallpox. How long would they live? That's a question that arises a lot, for example, in people who try to calculate insurance tables. When you say, okay, if you have a certain pre-existing condition, what rate should I price my insurance at for you? In order to do that, I must have to know how long you would have lived without that condition, as well as with that condition. So understand now what is the severity. If your disease does not affect your longevity at all, I can price my insurance to you at the same rate as I price it for anybody else who's completely normal. So he wrote down a mathematical model which we won't describe. Just remember the name. He comes from a very illustrious and famous family of mathematicians. This gentleman called Hamer, is later 1906, studied the epidemic curves. Measles is a disease that comes and goes, it ebbs and flows, certainly at that time. And this is, in a sense, the simplest of infectious diseases. It's transmitted from person to person. It's highly infectious, and it's also treatable very easily by vaccination, which the problem is that people don't take the vaccination. He introduced the idea that epidemics end when pretty much everybody who could have caught it has actually caught it. And it's an idea that has to be revised a little later. He described waves of measles, that is how measles comes and goes, as I said, in a periodic manner. And he was the first to use something called the mass action law, which we'll describe in the next lecture. So this is what I meant by periodic. So this is between the years of 1945 and 1965, 45 here to probably 70 here. You can see these very nice peaks. So this is the number of cases of reported measles in the United States as a function of year. So if you look at this, almost every year there's a peak. So what gives rise to this extreme seasonality, this extreme fluctuation here across these different countries in outbreaks of measles over here? That's something that Hamer was trying to understand, which we will also deal with as we go along. Now here is a famous name, and that's a famous name associated with India, and that's Ronald Ross, who won the Nobel Prize in 1902 for the life cycle of malarial parasite. He was actually born in India. He was born in Almora, educated in England, joined Indian Medical Services in 1881. But it's in Bangalore that he made the connection between water and mosquito control. So the sequence was in 1895, he found the first stages of growth of the parasite in the mosquito. In 1898, he found the life cycle of the parasite and returned to England in 1899 and published a bunch of papers at this point and a little later. Yeah. 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 Is it? Yeah. So it doesn't help at this point, but the question is what mathematical models of disease are consistent with a periodic outbreak. That's for example. Of course, now that you have vaccinations against measles, there are no cases at all and no outbreaks and so on. But in order to understand how this could arise and thereby make useful models for other types of diseases, it's important. This is just an experimental observation. This is a clinical observation of the repetition of number of cases on a regular basis.
So by seeing the year-wise data, you can also account for the seasonal variation.
Yeah, so there is a seasonal variation, and we'll come to how that is modeled in terms of what parameter is used to model this particular change. So he wasn't a very nice man because he sort of fought with an Italian called Grassi, and Grassi pretty much had done everything that Ross also did, but correctly identified the species, the life cycle, etc., etc. But Ross made sure that he never got the Nobel Prize, and this is a source of great tension between both of them. So, although Ross got the Nobel Prize for the life cycle of the malarial parasite, Ross always thought that his most important work was mathematical epidemiology. So he wrote down a couple of simple models for how vector-borne diseases work. And some of these he explained in a book called The Prevention of Malaria in 1911, etc., etc. And this was extended by MacDonald in 1957 to what's called the Ross-MacDonald model for malaria. But the most famous model of infectious disease was developed by two British scientists, Scottish scientists. One of them had an Indian connection. And for many years, the importance of this model was not recognized at all. This model is called the SIR model. So who are the people responsible for this? And that's these two people. So one person is called McKendrick, McKendrick was born in Scotland, trained as a doctor, but then joined the Indian Medical Services. Many doctors did at that time. They wanted to come and make their money by moving to the colonies. He was the director of the Pasteur Institute in Kasoli. Okay, so he spent many years in India, then returned to England in 1920, and then became superintendent of the Royal College of Physicians Laboratory for a period of about 21 years. This is in Edinburgh, in Scotland. The other person is called Cormac, and Cormac was trained as a mathematician, but got a job as a chemist. While working as a chemist in the same place in the Royal College of Physicians Laboratory, he had an accident that blinded him. As you know, for a chemist to be blind is almost completely career-ending condition. But Cormac had the advantage that he was an extremely natural mathematician. He would work out everything in his head. He didn't even have to write on a piece of paper. So Cormac and McKendrick got together to think about diseases. And the fact that sort of Cormac could work out things in his head without even worrying about paper, etc., ensured that this collaboration was a particularly successful collaboration. And that's when these two people wrote out the model called the SIR model. So one of the tests that they did for this model is data on the Bombay plague outbreak in 1905. Okay, so this is a very standard epidemiological plot that shows the number of infected people at time t as a function of time in weeks. Okay, you can see that at this point here, 0 weeks, there are no cases. The week they start, 0 weeks with respect to some starting point here. It rises up, reaches a peak, and then comes down, tails off over this. So the outbreak was over by about week 30, by the time there's basically 0 or 1 case out of here, which is around July 21st, 1906. And the whole period took about half a year, which is about 1 week to about 29, 30 weeks here approximately. And by this time, a certain fraction of the population had already been infected with the plague. So what's the job of a mathematical model? The job of a mathematical model is to try and understand data like this. How do you understand how this takes off here? How do you understand what the peak value of this should be? How do you understand how it falls down at the tail on the other side? So that's when Cormac and McKendrick said, look, this is data. This is nice data. Let us apply our theory, our SIR model theory, to this particular problem. So this is a comparison between the prediction of the SIR model by Cormac and McKendrick, this line over here, and these dots here are the same points that there were in the previous plot. And you can see that for a mathematical model, this does surprisingly well. It captures all of this increase out here, captures the fact that there is a peak. It also captures the decline at this point. This mathematical model is particularly simple and it has only one parameter. So there's really one parameter that goes into determining the shape of this particular curve. And of course, had it not been so good, there would still have been information in this. Of course, because in order to find these black dots here, you have to rely on people reporting disease. Often with diseases like the plague, there are many complex social factors involved. Nobody will want to admit that there's been a plague case in their house for fear that they will be taken quarantined, taken away, separated from their children, etc. So usually diseases tend to go underreported, especially diseases of an epidemic quality here. So there must be very stringent reporting requirements in place to ensure that you are actually counting all the cases. Remember, this is Bombay in 1905, and Bombay is a city where many people come from many different parts of India. They come from villages in the north, from the south, all over the place. So what do you do when you fall sick and everybody is falling sick around you? You go back home. And so you may not die in Bombay and be counted as a disease, as a death in Bombay or infection in Bombay, but you will be counted once you go back home. So you won't turn up here. So again, the tendency would be to underreport. Or you might have regions that are particularly well served by doctors. So in particular, let's say Ghatkopar has lots of doctors who are conscious of how to find out about plague. So they will report very well in that area and worse in other areas. So all of these are important to understanding what goes into these points here. And that's again important to stress that however good your mathematical theory is, however nice your formula looks like, however complicated it is and elegantly derived and so on and so forth, you're always confronted with the fact that you must relate this to real data. And there are many uncertainties in real data, and it's these uncertainties that you must also figure out a way to deal with. And now you would approach this from a Bayesian point of view, where instead of saying this is exactly the number that is measured, you can say that, look, this is the number of cases that are reported, but I assume that there is some probability distribution of a number of cases at this time, and some fraction of that probability goes into the reported cases that lead to these points here. So that's just an example of how our understanding of diseases and how to understand the many complicated factors that have to deal with how the nature of reporting, the nature of data, the nature of input to models must actually be accounted for. So as I said, Kermack and McKendrick invented the SIR model for diseases, and that's a model that we'll study in the next couple of lectures. This is really the cornerstone of what are now called compartmental models for disease transmission, how people get disease. And as I said, these people wrote this model down in the 1920s and 1930s. I think the late 20s, early 30s when this model was written down. And then everybody forgot about it for many years. Until finally it was Anderson and May in the 70s, Robert May in the 70s, who said that, look, this is an extremely important model. These people had understood the basics of how to model infectious diseases 40 years ago, 50 years ago, whenever it was, and they contributed to popularizing the original contributions of Kermack and McKendrick. I think they had probably died by then, so it wasn't much use to them. But certainly now we know what the nature of that model is. And it's a very pretty model. And so I'm going to enjoy teaching you about it in the next couple of days. And we will study this particular model in some detail as we go on. Okay, that's my last slide. I have now lots of time to answer questions. I have at least 15 minutes before tea is served. So please go ahead and ask me anything that you might want to ask me. Unless everybody understood everything.
So the fit model that you showed was after the fact.
Yeah. Yeah. So they had the data and then they said that, look, let's try and see whether the shape of this data can be accommodated in the analytic form that they had written down. So this is an approximation on the original SIR equation. They weren't doing it numerically. They found an analytic solution for SIR and they were able to justify it in terms of the etiology of the disease. So it's that sort of Gaussian peak, which is not exactly a Gaussian, but a sort of cos hyperbolic something or the other that actually gives, that they were trying to do. And finally it's a fit, but it's a one parameter fit, which is the R naught that comes in. And all said and done is actually fairly. This is supposed to be the most reproduced figure in books on epidemiology. Yeah. 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 The period, the sort of period in which you fall ill for plague is actually fairly short. So it's not as though you have been sick for a long time and then finally decide to go and see a doctor. It's typically 2 to 3 days. So that uncertainty is accommodated in here. And very quickly you can diagnose as plague. You can diagnose it very quickly as plague because you develop these buboes. So that's there are some nice books about the Bombay plague, which sort of describes some of the history of this. Any question? One question, two questions? So let's just see what all we covered. Go back to the beginning. Yeah, okay, I think stuff was actually fairly clear. I didn't think there's anything very complicated. If you have anything, you can ask, but you'll have to wait for about 15 minutes for tea to come. If you have no questions. If you have questions, I'm of course happy to answer them. No, because the reasons for that are much more complicated. It's much more individual. Yeah. So it's unlikely that you will be able to get a compartmental model because there is Compartmental models are sort of easiest written when there is an external causative agent that makes you go from susceptible to infected. The idea is to, we'll come across this later, but the idea is to divide the population into susceptible, infected, and recovered. And usually the transition between susceptible and infected requires an infectious agent, which is one of the microorganisms that I told you about, a bacterium or a virus or something. For non-communicable diseases, there is no single agent. It often has to do both with genetics as well as with lifestyle. With genetics, the issue is complicated because there's no guarantee that you will fall sick, because your body functions or the state of your health is a combination both of your genes as well as your environment. All of us may carry various genetic predispositions, but we may not actually fall ill with any of those particular diseases. And exactly what are the small changes in your DNA with respect to other people that ensures that you have more of a chance of falling prey to obesity or diabetes, all of that is not known. The other thing is that for every individual it's different. For example, usually it's a bad idea to have large amounts of carbonated fizzy drinks like Coca-Cola. And that's because it interferes with your metabolism in various ways. It may lead to some later susceptibility to diabetes and so on. But that can differ from people. Some people hate Coca-Cola, some people need 2 liters of it every day. So that at a very individual level, there are changes that is impossible to categorize as a simple susceptible and a simple infected. So usually their statistical models are more helpful to try to identify what are the determinants. So if you look at a large cohort of people, some of whom are ill, some of whom are not, so if you look at them forward in a prospective study, you can try and identify what are the important features that led to some of these people falling ill with a non-communicable disease and some of them not achieving that. But it has to be a combination of environment, circumstances, as well as genetics, which makes those issues a lot more complicated. It's not simply getting infected with something. Yeah, so the nice example of that, which I won't deal with, is something called the foot and mouth disease epidemic in the UK in the early 2001 or 2 or something like that. So foot and mouth disease is a disease of farm animals, typically of cattle, and it leads to the death of the cattle, and it's also communicable. It's an infectious disease that can move from cattle to cattle. It's important because dairy farming is a very important part of modern farming. And if all your cattle are going to die immediately and convey the disease to other cattle, that's going to devastate your own cattle industry, the dairy industry that's built around that. So when this was first diagnosed in the UK, the government was faced with this unusual question: what do we do? First of all, should we just kill all the cattle who have been diagnosed with? The technical word is cull. It sounds better than kill, but it sounds kind of the same. Do we cull all the cattle who we have been diagnosed with foot and mouth disease? Or do we look at a larger, because we know that cattle can transfer the disease to other cattle. And maybe this particular cow is ill but has not shown symptoms of being ill yet at a level which you can detect. But if you look at that cow or bull for about 1 week, then they will definitely fall ill. Now you can't kill every cow and every bull in the country, because that would devastate your economy. So then the question is, okay, suppose I tell you this farm has this case, 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 as of today. You know that you have to cull cattle, but exactly where do you draw the line? Shall I say that every cow within 1 kilometer of the farm where you reported a case, I will cull those cows? Or should it be 10 kilometers, or should it be 20 kilometers? What happens to the fact that cows can move between farm and farm? What do I do for that? So in order to address this, the British government set up a bunch of people. They essentially set up 2 or 3 different groups that really devoted themselves full time to modeling this and to providing the government with information from that modeling. And so they would all meet together once in 2 weeks. They would fly in from Edinburgh and various other places, meet with governments. Say, this is our current understanding of this is what we predict for the next week or 10 days as far as this epidemic is concerned. So then they said they managed to finally converge on a recommendation that said, for every farm where you have a reported case, cull within, I think, maybe 10 kilometers region, prevent motion of cattle from farm to farm, and that should be enough to solve the problem. So finally, a large number of cattle had to be culled. The cost was in the billions of pounds, but it wasn't near killing every single cow or bull inside the UK. So then that's an example of where a model was developed. That model was useful for policy. People acted on that policy in terms of actually killing farm animals, and that policy was successful in the sense that it led to a complete control of that epidemic in the time that came forward. So people, the moment there's a reported outbreak of disease, immediately WHO, 20 groups around the world are immediately saying, all right, here are the cases. So what we know about the disease, this is what our model prediction is. So side by side, everybody is racing. People are racing to sequence the organism. If it's a new virus, where does it come from? What might be the origin? What is the zero case? What is the index case from which it all started? Where is that index case from? What are the contacts that people had who have been affected by the disease? Who do they talk to? Who do they interact with in the weeks or days that led up to their being diagnosed? All of this, people are working constantly together exactly to do this, to decide what is good policy, what should you do. Even with Ebola, for example, a large part of dealing with Ebola was to understanding how West African burial practices were responsible for the spread of Ebola. In West Africa, like here, you tend to spend a lot of time physically very close to the person who is dead. You sort of wrap them up and tie them, and that gives you a huge possibility for contact with the body fluids of the dead person. With Ebola, you cannot do that because Ebola is transmitted through bodily fluids and it's highly infectious. Even very, very small quantities are enough. So understanding how local cultural practices, in this case associated with burial, actually influence the way the disease spread between people was a big part of it. So it's not just mathematics, but also understanding the social context and social practices that help you deal with the disease.